Welcome, everyone. I hope you are having a wonderful Monday. So today we're going to focus on viruses, but just to kick things off, I want to say just a quick few words about horizontal gene transfer, because we left last time without completely finishing that topic. So one of the things that we talked about at the end of class last time is that when we talk about genetic change, which is the major driver of evolution in bacteria and in all forms of cellular life, and in non-cellular things like viruses as well, universally there's a mutation, which is when the template DNA that's copied prior to cell division acquires errors, and those are passed down. But these other methods of gene change happen in bacteria frequently: transduction, conjugation, and transformation. And these are somewhat unique to the prokaryotic space, and they drive faster evolution. And so I just want to remind us of what those things are. Transformation is what we've done in the lab, right? We did this in lab, and we're going to collect data this week. Basically, this is when a cell acquires DNA because it just wanders across it. Some DNA is lying around naked, not enclosed by a cell. And this is because the bacteria that used to have the DNA has died. Conjugation requires living cells to contact each other. And transduction is when that exchange of DNA occurs because of a viral intermediate. We talked about this iconic transformation experiment. And then I just want to make sure that we understand that conjugation happens differently between the gram statuses, that gram negatives use a fertility polis and gram positives use a mating bridge. And as a consequence, you would not get conjugation between gram negative and gram positive cells. You also want to make sure that for the test, you remember who's who. Gram negatives use the fertility polis and gram positives use a mating bridge. And then this is really relevant to our conversation today about viruses. This is a description of one of the ways that you can get transduction. Okay, so here's the example. We're going to see another version of this today, which is actually the more common way. But just briefly, a virus injects its DNA. That DNA causes the cell to replicate more virus particles. In the process, some bacterial DNA might accidentally get inserted into the capsid so that when it goes to its next host, it injects the bacterial DNA instead of viral DNA. And then that bacterial DNA could be inherited by subsequent generations. Okay, so one of the things that we may or may not get a chance to do today is a genetics Kahoot. I'm not going to do it right now. If we have time at the end, we'll do it, or we will have it at the end of class on Tuesday. So with that, I just wanted to switch to our main topic today. And let's get rolling. Viruses, guys, viruses. So there's so many things we can talk about when it comes to viruses. What makes them viruses? What are those specific structures and features that they have? How do we classify? How do we grow them? How do the ones that infect bacteria replicate? And then, of course, how do animal viruses replicate? Because there are some pretty major differences having to do with the differences between the host cell types. This we're going to reserve for Tuesday. Okay, so we'll talk about this tomorrow. But we will just today discuss some of the things that are not viruses. Just to warm up and kind of ground ourselves in things that are familiar and interesting. I'd like you to think about any famous viral plagues. It could be recent, it could be historical, ancient, that have plagued humanity. Some disease that was caused by a virus that has caused a lot of suffering and death. Put that into the chat and I'll read out your answers. Yeah, the Spanish flu from the turn of the last century, polio, Ebola. Coronavirus. Yes, some great examples, guys. So some of the deadly viral plagues that are sort of most famous in microbiology, smallpox is always front of mind for two reasons, or three really. It's been in the human population for as long as we have written records, like ancient Egyptian hieroglyphs depict smallpox victims. And at the same time, it's one of the most amazing turnarounds, I guess, based on modern medicine. We were able to develop a very effective vaccine that rid the world of this disease that had been with us for millennia. It's caused a lot of suffering and death over our history. And it also has quite a high case fatality rate. This is a metric we'll talk about in Unit 3, which has to do with how likely you are to die if you get sick. So 30% is really high. The only other ones that are kind of similar in that sense are things like Ebola is higher, and sometimes newly arising viruses in the very early stages of them showing up in the human population can be high. But this is really high, especially for something that's been with us for a long time. And it's also had major historical impacts as a result of migration over historical time. So for example, the First Nations people in this area of the world in North and South America experienced a great deal of suffering and death when they first encountered smallpox, when Europeans migrated to this continent. And the vaccine, which has basically reduced cases to zero, is estimated to save about 5 million lives a year. Some of you mentioned the flu of 1918, shortly after the First World War. It's estimated on the upper edge of the estimate, 50 million people died of this flu worldwide. And you can see that in contrast to smallpox, the CFR is 10 times lower, yet this is quite high still, especially for an extremely contagious disease. And so this is reflected in the numbers of people that died in just 2 years. It was 2.5% of the global population. And currently, we're obviously in the middle of another famous viral plague. About 6.3 million deaths conservatively have been confirmed, at least 520 million cases. The CFR is, to calculate this, you take confirmed deaths over confirmed cases, and it's about 1% at this time, but it varies from region to region. There's a link if you're super interested to go look at. None of you mentioned measles. That's another famous one. It has been greatly reduced by vaccination, by effective vaccines, though not eradicated. Just in comparison, in the 80s, when I was going to high school, about 2.5 million deaths worldwide every year, annual deaths. And in 2018, more recently, the numbers have gone way down, although it's still significant amounts of people, and most of those are like infants and toddlers. The estimate is that the vaccine prevents 2 million deaths a year or so. Polio, some of you mentioned polio, has also been greatly reduced by a vaccine, though it's not eradicated. The peak in the United States occurred in the mid-50s, and there were tens of thousands of cases, thousands of people died. The CFR is very high. But the vaccine really just made a huge difference, brought domestic cases to zero. It's still endemic in a couple countries in the world, but the use of this vaccine prevents several million deaths, has prevented several million deaths over the last 2 decades. Okay, none of you mentioned the bubonic plague or the Black Death, but that's something that some people often think about. This is not viral, and that's why I mention it, because sometimes people think of it and they think it's viral. It's actually caused by a bacterium, but similar to smallpox, has been in the human population for a long time and has caused a lot of death. Like a third of the European region, people died in just a couple of years in the last major outbreak. As many of you now know, it's caused by a bacterium that's spread from rodents to people by fleas. The worst version of this for us is the pneumonic version, when it gets into the lungs, and then it becomes person-to-person contagious, which is why some people think it is viral. It's been well controlled with antibiotics and pest control. Okay, so another little warm up, a little grounding. I'm going to have you do some Googling. If you are driving or not in a safe place to do this, please do not participate. I don't want anyone to die on my watch just for answering some questions. But right now, if you have the ability, I'd like you to look up, Google the size of the viral particle that causes COVID-19, the SARS-CoV-2 viral particle. See if you can find its size and post that in the chat. And then I'll wait to see what your answers are. And if you are not in a position to Google, but you can just put a symbol into the chat, you could put a question mark to let me know that you're not able to answer. And if you can't do that, just don't do anything unsafe, please. That's fine. I just want to make sure I'm not stealing anyone's thunder who has an answer and wants to share it. Yeah, great. So as many of you have discovered, the size of this virus is very typical. It is about 100 nanometers in width, or 0.1 micrometers. That's quite typical for viruses. Some are a bit bigger, some are a bit smaller. What do you know about the symptoms? Put that into the chat. What are the symptoms? Most of us don't even need to look this up, but you might want to, just to see like if there's some fun ones that aren't as common that you want to share. You could look it up and go, what do people say besides the usual things? Okay, so dry cough, cold and flu-like symptoms, fever. So GI, things like nausea, vomiting, diarrhea, respiratory things like shortness of breath and the cough. Yeah. Okay. We will, yes, and hypoxia, not being able to get enough oxygen in the blood, loss of taste and smell, that one can persist for a long time, having a headache, indeed. Good. Okay. And fatigue, that's right, Lori. Yeah, no energy. What about the tissues in the human body that are actively infected by the virus? What tissues can it get into? Put that into the chat. You can have a little Google for that too. Put what you know, put what you find. Yes, so one of the things that it can actively infect is your gastrointestinal tract. So that that tube in the middle of your body that food passes through, right? So the tissue lining it is susceptible to the virus. That is one thing. It's true. Anything else? Yeah, the lungs and the throat. Anything else? Your blood vessels. Yeah, your respiratory tract and your blood vessels, your vasculature, indeed. Indeed. Any other ones? Yeah, your liver and kidney and brain. So there's several internal organs as well as, so there's the respiratory tract. That's sort of obvious, I think. Most of us already aware of that. The GI tract, not as commonly known, but still some people recognize that. And then some of our internal organs. All right, what is the protein receptor on these cells of these tissues that the virus uses to gain access to your body? What is the specific protein receptor on the surface of the cells of your human body that the virus latches onto. This one you can find. Some of us just know it, but more common, people need to look this up. Right. Mm-hmm. 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 Yeah. Yeah, that's right. It's called ACE2, and thank you for also finding what it stands for. ACE2 stands for angiotensin converting enzyme 2. All right. So I asked you some questions, some fact-based questions. And just to be very clear, let me go back here. I would never ask you to memorize these, right? If I wanted you to know this, I would provide this information on the test. Although I would expect that you would know what the range was likely to be, right? But I would give you this information if I wanted you to have it. You don't have to memorize that. But with that information, right, now that we have it, can you see the SARS-CoV-2 particle with a light microscope? Use your yes, no tool to answer, or you can put your answer into the chat, whatever is easier for you. Right. Yeah. So if this was a board meeting, the no's have it. The no's are correct. Why? Because it's so small, right? Below the resolving power of a light microscope. We talked about these symptoms of infection already, right? Some flu-like symptoms, some GI symptoms, nausea, vomiting, diarrhea, fatigue, hypoxia, and coughing, other symptoms of pneumonia, but also in a more severe case, organ failure and death. We talked about this also, the receptor, the cell receptor used by the virus to gain entry is called the ACE2 receptor. And shocking, it's found on all these tissues that kind of correlate with these symptoms. So it's found on the cells of your GI tract, respiratory tract, and several of your internal organs in your vasculature, your bloodstream. Now, are there other things that could cause these same symptoms? Of course, there are many other microbes, bacteria, fungi, other viruses. So based on that, can you diagnose COVID-19 solely based on symptomology? Everyone's instinctive response was no, and everyone is correct. This is always the case, right? All right, so let's move on to the size. I asked you to look up the size of coronavirus. We talked about how you can't see it on a light microscope. Viruses are much smaller than cells. Let's start there. But when we look at the range, it's quite broad. So some viruses are so small, they're the size of ribosomes, and others are so big, you could almost see them on a light microscope. Still would be too skinny, so you probably wouldn't see it, but you can see a kind of a variety of sizes and a variety of shapes as well. And one of the things that is so fascinating about viruses is that, as we've just discussed, you cannot see them with a light microscope, which means that we had no way to visualize them until the 1930s when the electron microscope was invented. And yet people knew about viruses, their existence. They couldn't really explain exactly what they were, but they knew they existed because of filtration. Now, filtration, as it's practiced in the modern age, is a way to sterilize things that can't be heat sterilized. The most typical way to sterilize something for safety is by running it through an autoclave. Like all the media that you're handling in the lab, the sterile media has been put through an autoclave, and that's just a sealed chamber with high pressure and high heat. high pressure steam heat. That's what's used in hospitals to decontaminate and sterilize equipment that's going to be reused. But there are some things, for example, nutrient broths, like maybe there's some specific vitamins in here or something of that nature that cannot be heated. And so to make those safe for consumption, we pass them through a filter. The filter itself is just a piece of special paper with holes in it that are the size of about 200 nanometers. That's the diameter of the hole. And the filter itself is sterilized. This whole plastic doohickey is created in a sterile way. So then you just pour your liquid over the top and the filtrate comes through and it holds back cells like bacteria and parasites and things. But it doesn't hold back viruses. Obviously, they're too small. They just slip through those holes, don't they? And so long ago, well, maybe not as long ago as one might think, but people were doing these kinds of things in a more rudimentary way to filter out bad things. And some of the time, whatever the infectious agent was that they were filtering out was not cellular, but viral, and it would pass right through and the filtrate would still be contagious. And so people started trying to figure out what that was being passed through. Now we know it was just a small particle. Okay, so having done your prep, you know some things about viruses. What are some of the things that are defining for this group of microbes? Write them into the chat. I will read them out as you write them in. What makes a virus a virus? Their genetic material is DNA or RNA, but never both. They need a host cell in which to reproduce. They're considered to be non-living. They're not cellular, that's for sure. And they're given this awkward descriptor, obligatory intracellular parasite, which is just a fancy way of saying they have to be inside of a host cell in order to replicate. A few other things. So they have to be inside of a host cell in order to replicate. They have a genetic material that's either DNA or RNA that is surrounded by a protective cage made of protein. Some viruses additionally have a phospholipid bilayer wrapped around that called the envelope. And then there are attachment proteins. Depending on the type of virus, they might be called spikes or tail fibers. Tail fibers is the term used for bacteriophage and spikes for animal viruses. And most are quite specific in their host cell type that they can infect. For example, if you have a cold, you are probably never worried about your skin catching a cold because the cells of your skin are too different. And the receptor that the virus can enter is on your nasal epithelia and your respiratory tract, but it's not on your skin. And similarly, a lot of times viruses are so specific that even another species with similar organs would not be infected because they don't have the same exact receptor. This leads to having a very limited host range. Okay, so as we discussed, the virion is made of genetic material, protein, sometimes lipid. What is the RNA or DNA used for? Put that into the chat. That's right. It is the genome. It is the instructions for the viral proteins, instructions for making viral proteins. Just like your genome is the instructions for making human proteins, the genome of a virus, whether it's RNA or DNA, is the instructions for making viral proteins. What viral proteins? What does a virus use protein for? Put those into the chat. Yes, Ayumi. Viruses have, remember, a protein capsid, a cage of protein around their genetic material. They need the proteins to make those, and they're not human, and they're not going to, their host cell doesn't have those genes. So what else? They need the capsids that are made of protein. What else do they need that's made of protein? What do they use to grab onto their host? Yeah, spike proteins, attachment proteins. And some of the time, depending on the type of virus they will additionally need enzymes, specifically polymerases, to make more RNA or DNA. But a lot of times they don't need those enzymes, especially if the host cell already has the right enzyme for them. Why do some viruses have a lipid component? What is that lipid component part of? Ayumi asks if protein coat is equivalent to capsid. Yes, the capsid is made of protein, and it is sometimes called the protein coat. The capsid is the protein coat. They are synonymous. Why do some viruses have a lipid component? What is it called? What's it made of? Where did it come from? Yeah, it's a phospholipid bilayer. It's called an envelope. And how did it end up on the virus? They got it from their previous host. They wrapped themselves in the skin of their last victim, and they're going to use it to sneak into their next victim. Okay. So here's a little cartoon of a common type of virus, and the shape looks sort of geometric. The subunits are capsomeres, and you just have 1 or 2 or 3 proteins that all together fold up into one little bead or one little brick, and then you take a whole bunch of these bricks and you just build yourself a little cage. And that is built around the genome, which is either RNA or DNA, but never both. And then there's spikes or attachment proteins in the capsid. For those viruses that have an envelope instead, the spikes are in the envelope. So we can see that here. These are typical animal viruses, and they're naked or enveloped, meaning do they or do they not have this external phospholipid bilayer? Okay, when you look at these two pictures, we can see genome, we can see a protein coat. Additionally, this has an envelope. On the very, very surface are these spikes. What is it in the structure of the virus that most directly determines host specificity? What in the structure of the virus limits it to being only able to get into specific host cells and determines what those will be? Yeah, good. So we have a couple of good answers in the chat. There are two sides of the same coin. So protein receptor on the host cell, attachment spike on the surface of the virus, they connect together in a very specific way. But what I think is a helpful analogy is to think about these viral spikes like a key that only fits into a specific lock to unlock that cell. And so it's not going to be able to put its key in a different cell that doesn't have the lock on it. All right, virus morphologies can come in a couple different flavors. Most common is this one. What's it called? Where you have a bunch of bricks and you make a little triangle out of them, and then you make another triangle and you make another triangle and you just put it all together. What is that called? Polyhedral. If it looks like a geodesic dome, polyhedral. Isometric is the other word that's commonly used to describe this. And the most common is an icosahedron, which has 20 sides, 20 triangular faces. Now over here, we see this so-called helical morphology of virus, where you just build a circle and then you keep adding and making the circle taller and taller and stuff the DNA or RNA inside. And then we've got complex viruses that have a little bit of column A, a little bit of column B. This is typical of the bacteriophage. So let's have a peek at that bacteriophage. What it is grabbing onto the cell with is not called a spike, it's called a tail fiber. And part of that distinction is not just arbitrary, but the job of this virus is a little bit different than the job of the virus that's trying to penetrate a human cell. Because what animal viruses do is grab the plasma membrane and using one of the cellular receptors on the membrane. Is that what bacteriophage do? What are they grabbing? Bacteriophage grab bacteria to get in. What are they grabbing specifically? Are they grabbing the plasma membrane? What does the surface of bacterium look like? Is the plasma membrane the first thing you encounter? What do these bacteria have to grab onto and navigate past in order to get inside the cell? Okay, maybe the glycocalyx, but not all species have one of those. It's not going to be the fimbriae. What is on the surface of every bacterial cell? Every single one. Flagella is optional. For some species have it, some don't. That does not determine whether or not you can be infected with virus. What's the wrapping around the bacteria that has to be navigated by this virus? Yeah, the cell wall. So how this works is really cool. You can see it happening up here in this electron micrograph. But the virus grabs on with its tail fibers. Sticky tail virus grabs the cell wall. Right here, there's an enzyme called lysozyme that we've talked about before. Lysozyme chops up peptidoglycan. So it chops a little hole, like a little ice fishing hole in the cell wall. And then this shaft contracts like a hypodermic syringe to inject the DNA through the wall and into the cell. Now, here's some information you do not have to memorize, but just to kind of, again, give us a sense of, we've learned what viruses are like. Now we can sort of take information and slot it into the different parts. The size we've talked about, it's a polyhedron, it's enveloped, so it has a phospholipid bilayer on the outside. It has a small RNA genome that encodes, among other things, the S-glycoprotein that binds the ACE2 receptor on epithelia. And here's all the tissues where ACE2 is expressed. So just getting back to what that DNA or RNA in the virus encodes, somewhere between 10 to 100 genes. Now, if we think about cells, like human cells, 30,000 genes, bacteria even like several 3,000 to 4,000 genes. So this is much, much smaller. Viral genomes, in addition to being small, are not always DNA. Sometimes they are. Sometimes they're RNA, and if they're RNA, they might be single-stranded, and if they're single-stranded, they might be positive sense or negative sense. They could be double-stranded, and they could be retroviruses. So that is all important, and we're going to cover that tomorrow. It matters for replication and transcription. We've talked about this already. What's the genome going to code for? The capsid proteins, attachment proteins, and sometimes viral enzymes like polymerases that are required to replicate that particular virus's genome. Classifying viruses is a fun hobby. We can classify them based on their morphology. What were some of the options we mentioned? Throw those into the chat. Any one of the ones that you remember? There's 3. What do we call it when it looks like a geodesic dome? What do we call it when it looks like a chimney? What do we call it when it looks like a little bit of both? Put any of those terms into the chat. Yes, when it looks like a geodesic dome, we call that polyhedral. Another word for that is isometric. And the most common is the icosahedron with 20 sides. The one that looks like a chimney, we call it helical. And the one that looks like a bunch of things, we call that complex. So we could refer to viruses based on their shape. We can refer to viruses into categories of naked versus enveloped, meaning do they have the envelope? And we can also often refer to them based on their genome structure, which was, they could be DNA viruses, they could be RNA viruses, they could be single-stranded RNA, they could be double-stranded RNA, they could be positive sense single-stranded RNA or negative sense single-stranded RNA or retroviruses. And we also can classify them based on how their genome is organized, either in one molecule or multiple molecules. Now, again, I'm going to show you some things that you do not have to memorize. We're just trying to ground ourselves in this material. And so norovirus, I'm sure all of us have heard of, is transmitted by ingestion of saliva, feces, or vomit. It is common in crowded conditions, in particular cruise ships where people are drinking reused, recycled water, and is passed by contaminated surfaces and food and water. And so this is an enteric virus and infects the GI tract. The route of transmission is fecal oral. What about this one? Put this into the chat if you know what. Influenza virus transmitted by exhaled droplets when you sneeze or cough is considered what category of virus? It's not enteric, it is respiratory. That's right. Respiratory viruses, the route of transmission is droplet transmission. HIV is transmitted when mucous membranes come in contact with bodily fluids, either direct contact with another mucous membrane or contact with semen, breast milk, blood, and other bodily fluids. That might happen during penetrative sex, during childbirth, if you receive a transfusion of contaminated blood, or during reusing needles without sterilizing them in between. What classification do we give this? Not enteric, not respiratory, but what blank virus? Sexually transmitted, right? Yes. Although I always feel like that is somewhat misleading because, you know, childbirth is not sex, transfusions and needle reuse is not sex, but it is a very intimate contact in a way, right? Okay, rabies, transmitted by the bite of a rabid animal. What do we call this type of virus? That's right. Exactly, zoonotic. So you have to understand these terms and be able to extrapolate from information given to you, but you're not required to memorize norovirus details or influenza virus details. I would provide that. And so that same thing goes here. We're just gonna check in what we think about this. Each of these I'll ask you for your opinion, but you don't have to memorize these. But measles, which of those classes, enteric, respiratory, sexually transmitted, zoonotic, applies to measles? If you don't know, put a question mark. And you don't have to know. It's not in my curriculum. And you don't have to know it for the test, but let's just see if you do know, because that's fun. If it's any consolation, several of these, when I started my studies as an undergraduate student, I had no idea. But those of you who have worked a little bit in healthcare may know. Yeah, so measles is a fun one. Measles is respiratory. That's right, respiratory. What about chickenpox? My hint to you is it's not what I thought it was when I was young. When I was young, I thought chickenpox was passed by direct contact, like those little weepy sores that you get. I thought that you got exposed to those like on your skin and that's how it was passed. But no, it turns out it is what, guys? Put a question mark if you don't know. Just so you're staying alive, you know, keeping in the game. Yeah. Also respiratory, crazy, huh? Respiratory, measles, respiratory, chickenpox, respiratory. It causes a rash, but it's respiratory. That's how it's transmitted. Human papillomavirus, also known as HPV. Any thoughts on this one? Yes. It's in this classification of so-called sexually transmitted viruses, but it does require sort of direct intimate contact. Polio. This is another one I did not know when I started my studies. Polio. Some of you will know this. And the interesting thing about polio is when it was sort of at the height of infectivity, people did not really necessarily know exactly how it was being transmitted. Yes, this one is enteric. And it's passed by ingestion, usually of tiny bits of feces in contaminated water. Herpes simplex, this one I think is pretty, most people know this, another STI. And then West Nile. What's West Nile? Right. This one's passed by the bite of mosquitoes and usually from another animal. So yeah, West Nile is zoonotic. Okay, now when you want to grow viruses, and there could be lots of different reasons you want to study them or you want to make vaccines out of them, then you cannot just put them into a broth the way you can with bacteria, right? They need a host cell to replicate. So let's talk about bacteriophage, right? Remind us, what is the host cell for bacteriophage? Put that into the chat. You want to grow bacteriophage. What do you need to provide the bacteriophage? What do bacteriophage infect? What is their host cell? Put that into the chat. It is not eukaryotic cells. So what does that leave? Bacteriophage infect. Yeah. Bacteriophage infect bacteria. So what you see on your screen is a way that this is typically done. So we're focusing again on bacteriophage, which is just one of several types of virus. It's the one that infects bacteria. So here we see a plate. This is just like what you've been handling in the lab. The plate is opaque because there's a lawn of bacterial cells on it. In order to put cells onto this plate to grow that lawn, a thick broth was inoculated with a little bit of virus. And so a small number of cells receive the virus. When you spread those cells out on this plate and you let them grow up, any cells that were not infected are just going to grow into a lawn. But a single cell infected with a virus particle, when it lands on this plate, that cell is going to be taken over by the virus. The virus will replicate, explode, and spread to all of the neighbors. And those cells also replicate the virus, explode, and spread to all the neighbors. So this sort of circle of death expands from that center. It represents all virus particles that were derived from a single original virus. Okay, now when it comes to animal viruses, we still need to provide them with host cells. What are some ways to grow animal viruses? Put that into the chat. Yes, one way that's commonly done is to inject the virus into an embryonated chicken egg that has all of the right stuff for the virus to replicate. The most straightforward way would be to inject the animal host, right? Obviously, you need to give the animal virus animal cells, so you could just give some animals the virus. But there are several downsides to this approach. Can you think of any? Why would embryonated eggs be a better choice than growing the animal virus inside of an animal host as it's living? It's true that a living animal is not a very controlled environment, is it? What else can I think of as being like somewhat unideal? What if it's a virus that only infects people? Is everyone going to sign up for this? Will you sign up for this? Yeah, please infect me with this deadly virus or this uncomfortable virus, anywhere along that space. And there's also things to consider about suffering. Yeah, it could hurt or kill the animal, certainly. Ethics. So not ideal for human pathogens, not ideal if you don't like hurting and killing things. And just from a completely practical standpoint, very expensive. Now, this, much cheaper. Nobody gets their panties in a twist. No humans were involved in the making of these. Now, there is an issue, and a lot of times this is done for vaccine production because we need to make a lot of virus particles for the vaccine for relatively inexpensive. But you've then got to purify the virus away from the egg proteins, and that's not very easy because egg proteins are quite sticky. So what's the downside of this approach? Yeah, though it is essentially impossible to remove all of the protein from the vaccine, from the virus, purified virus. And so if you have a person who has an egg allergy, they cannot take this vaccine. For that reason, there are other methods that are sort of in between these two. This is more expensive and cumbersome, but much cheaper and less of a problematic than growing a living animal. This is to take primary cell culture from the animal, one cell type that you do know, and to grow the virus in that. And we also have some immortalized cultures that are made from cancer cell lines that actually can grow quite well. And so you can use this technique to make a version of the vaccine that people with allergies to eggs can take. Okay. So in terms of identifying viruses, there's a tricky thing, right? Identification is harder than with bacteria because you can't see it under a light microscope. You can only see it with an electron microscope, and even then it's tricky because they're so small. But you can start to identify that a virus might be present by something called cytopathic effects. And you can become more confident about who it is using serological testing. That's the basis of like the COVID test at home, the antigen test, and a variety of other similar tests. Or you can sequence the genome of a virus and really find out who it is. I just want to briefly introduce you to the cytopathic effects. So even though you can't see the virus under a light microscope, what you usually can see is that the cell is looking weird. Sometimes that weirdness manifests as like sort of gatherings of virus in the cytoplasm that you can't see the individual particles, but you can see like sort of these inclusions in the cytoplasm. Sometimes they show up in the nucleus. You'll see the cells exploding when they under conditions where they normally wouldn't, or fusing together. Here are many, many nuclei of some original cells that have fused together and also behaving strangely, changing their shape, changing how frequently they divide, being willing to crawl all over each other. You can see those differences, for example, in this picture here. So here's an electron micrograph taken of a cell type that is normal in one side and infected in the other. And this would allow us to determine if it was infected. Which is the infected side, do you think, A or B? Put that into the chat. If you're not sure, take a guess anyways. And again, you don't have to memorize this picture. You have to just understand the concept that it is possible, even though you cannot see virus with a light microscope, you could see this under a light microscope, this change. You'd be able to identify if something was virally infected or not. It is this side, okay? It is this side. But the most important thing is just to recognize that one can see a difference, even though we can't see the virus particles. We are taking a break, not until 11 a.m., because that would be a long time, but until 6 p.m. And then we will resume our conversation about viruses. I will see you in 9 minutes. All right, welcome back, everyone. Let's talk about how viruses replicate. So all viruses have a similar trajectory. They have to get into their host cell, they have to make more of themselves, and they've got to get out. So there's all kinds of details to learn that we'll focus on mostly tomorrow, but it's some variation on this theme. Now, if we look at the change in virus number in a typical infection, starting with the number of viruses that enter some particular amount of cells, let's say it's 100, then what happens initially in the early stage of the infection is that the number of viruses appears to go to 0. This is followed by a rapid amplification step where we see lots and lots of virus particles, and then they start to decline again. What is this period called? What is this period called? It's not the lag phase. It's sort of the equivalent, I guess. It's not really even equivalent, but yes, Haley. Haley is right. Everyone else have a peek at Haley's answer, then you can put it into the chat to practice your memory of this concept. That's right, Lori. Yes, Emily, you are correct. This is known as the eclipse period. I'm going to ask a couple people, if necessary, we'll start with one, what it means, like why is it that the number of virus particles appears to go to 0? Haley, can you explain that? You were the first to say what this is called. Do you know why this happens?
Yeah, so the eclipse period, I believe, is when all of the virals have entered into cells, and so you're no longer seeing them for that short period of time.
Yeah, that's right. So this is when the viruses have entered their hosts, and this is the part I think that is maybe an additional level of detail, but the viruses have entered their hosts, and even if you looked for them with an electron microscope, you wouldn't see them. Okay, that's how much they have disappeared. But it's not that they evaporated into thin air. When they get into their host, they take their coat off, that protein coat, that cage around the genome, they take it off. So now they're just a piece of genome, and you're never going to see that. So that's why it looks like there's 0 virus particles, because I mean, in some senses, that's true. but they've taken their coat off, and now the genome is beginning to boss the cell around to make more. And once the virus particles have been assembled, we start to see a rise in number. We're going to look at lots of different life cycles of viruses, but today we're going to get sort of the bacteriophage replication out of the way. So again, what is the host cell for bacteriophage? We covered this earlier. Let's see if we remember. What is the cell type that is affected by a bacteriophage type of virus? Exactly. Bacteriophage infect bacteria. Now, earlier we talked about how one of the defining features of viruses is that their genome is either DNA or RNA, and they never have both types. So this is distinctly different from cells. But in the case of bacteriophage, it's even more restricted. They are only one type of genetic material. So now you have a 50-50 shot of being right. Even if you don't know this, put your guess into the chat. Is it DNA or is it RNA that bacteriophage genomes are made of? Great. Okay. So regardless of whether you got the right answer or not, does not matter. What matters is that you remember it. And so the answer is DNA. Bacteriophage always are made with DNA. Okay. There's two subcategories of bacteriophage based on their life cycle, the lytic cycle or the lysogenic cycle. And we will talk about the lytic cycle first because it's more straightforward. To get in, the viruses attach and penetrate. We'll see exactly how that works. Then they make more of themselves. Then they explode the cell to get out. One virus goes in, like hundreds come out. When we're talking about bacteriophage, the attachment proteins have a particular name. What is that name? Spikes is a good guess. It's not spikes. That's the name we give for the attachment proteins in animal viruses. Bacteriophage, we give it a different name. It is tail fibers. So that's something to remember. Now, the other thing that's really different in this early stage of the infection is, as composed to an animal virus, is what exactly the virus has to get past. So a bacterium structure is different than an animal cell structure. What does this viral particle have to traverse or overcome? What is the obstacle preventing the DNA from getting into the cell? That's not found on animal cells. If you have no clue, put a question mark, but do put something. Right. They have to get their DNA past the cell wall. Now, animal viruses, they enter the cell whole. They bring their coat with them. Then they take their coat off. But bacteriophage land, they use their tail fibers to grab the cell wall. They use an enzyme to drill a little hole. They contract like a hypodermic syringe and inject their DNA through that hole. But the capsid stays outside. That's very different. So they use an enzyme to make a little hole in the cell wall, like an ice fishing hole. They inject the DNA through the wall, but the capsid stays outside. And then once that DNA is inside, does it need a viral enzyme to replicate, or does the viral DNA use the host's cellular enzymes to replicate? What do you think, viral or cellular? Again, you have a 50-50 shot. Even if you don't know this, you might as well take a guess. Great, yes. So the DNA, it's very straightforward in a sense, right? The DNA, to replicate and get more DNA genomes, you just use the host cell's DNA polymerase. To express that DNA into viral protein to make the capsids and the tail fibers and the lysozyme and all that, you use the host cell's RNA polymerase to make mRNA and the ribosomes to make protein. Now, how they leave is the lysozyme enzyme that's present in the original viral particle. It's also, the gene is also in the genome. So as the viral DNA is being transcribed and translated, lots of lysozyme is being made, and that lysozyme eventually explodes the cell. So we can see that in pictures. Tail fibers grab and attach to the cell wall. Lysozyme drills a little hole through the cell wall. The hypodermic syringe apparatus injects DNA through into the cytoplasm, where it is replicated by the host cell's DNA polymerase and expressed by the host cell's RNA polymerase and ribosomes to make more virus particles, which then, after the lysozyme that's floating around in here, explodes the peptidoglycan cell wall, and out they come. So that's the lytic cycle. What's the other version of a bacteriophage life cycle? If you don't remember, you can put a question mark. Do engage with the prompt. Yes. Good. It is lysogenic. Now, the lytic cycle is an acute infection that causes rapid lysis and death of the host cell, but the lysogenic cycle frequently leaves the host alive, maybe for generations. So we can see that we've added the lysogenic cycle to this diagram. Which side, the left or the right, represents the lysogenic cycle of viral infection? Yeah, it's the one on the right-hand side. So what happens? Here we can see the lytic cycle, right? We inject our DNA, the DNA is replicated, capsids are replicated, we get an acute infection with lysis, and off he goes to find a new host. In the lysogenic cycle, when the DNA is injected, and this is a species thing, right? Different bacteriophage species do this lytic cycle, other ones do the lysogenic cycle. The viral DNA first enters the bacterial chromosome, and it may stably stay there for several, even sometimes many generations. It's just inherited, passively inherited as the cell goes through cell division. So it becomes, at least temporarily and sometimes long-term, it becomes part of the bacterial chromosome. At some rate, some of the time, some of these progeny, the viral DNA hops back out and causes a lytic cycle. So one of the things that's important to note about this process is that this is the most common form of transduction that leads to important pathogenic outcomes. Okay, so you can see how this might lead to bringing DNA between bacteria. So let's suppose this guy injects his viral DNA, the DNA gets inside, it's now called a prophage when it's inside the bacterial chromosome, and it's just inherited, and after some point in time, the viral DNA hops out and causes this cycle. Quite often, when the viral DNA hops out, it's not a clean. It doesn't hop out like right at the borders where it was originally put in. It often will take some of the bacterial DNA with it, so that when this next step happens, we then have a species that is passing around a gene from this and injecting it to its next host. And what was the name of the DNA once the viral DNA is inserted into the host? What do we call it once it's inserted into the host? The DNA has a special name. Yes. It is called the prophage. Okay, so this method of lysogenic cycle is responsible for a horizontal gene transfer. And what do we call it when we use a viral intermediate to do horizontal gene transfer and transfer genes between bacteria? What's that called? And if you don't remember, putting a question mark is the perfect answer. Just put something. Yes. Lovely. I love all the question marks. I love all the answers. They're all welcome. Perfect. Yes, it is transduction. Now, I'm bringing this up, and you don't have to memorize these examples, but you do have to understand the concept. When transduction occurs by a lysogenic cycle, it's called lysogenic conversion. It's just a fancy way of saying that that the bacteria acquire DNA through a lysogenic cycle from a previous host. But this is the mechanism that explains how our current pathogen that causes botulism, Clostridium botulinum, got the botulism toxin gene, and it's the cause of the rise of diphtheria and this new form of E. coli getting a Shiga toxin gene from Shigella, and many other examples. So a lot of bacteria that are famous for causing terrible human diseases do so because they have particular toxins that they didn't invent, they got from somewhere else. Okay, so we're gonna do a little breakout right now. And you might want to take a screenshot of this picture of this screen, just so you know what to do. You're gonna be looking up some information about a virus, just like you did with me in the main class for COVID. But you're gonna be looking up one of these. I'm gonna put you in groups, and within your group, you're gonna have 5 or 6 people. You're each gonna pick one and look up this information and then report back to your group so that everyone in the group is aware about all this information, but you don't all have to look up everything. So if you had 6 people, let's suppose one of you would do HIV, one would do rabies, one would do smallpox, one would do Ebola, etc. So while I create those breakout rooms, let me tell you what it is that you're going to be looking up. Okay, you're going to be 5 to a room, so there might be one of these you don't get to. But you're going to be looking at the size, and you want to take notes when other people are talking, so you have the information that you looked up, but also the information they looked up. So you're going to find out the size. What type of genome is it? Is it made of RNA or DNA? Was it single-stranded or double-stranded? Is it minus sense or plus sense? Is it naked or enveloped? If you can find a picture, look at it, and just in your own words, describe its shape. And then if you can, find the cell receptor targeted by the spikes, mention that. Okay, so once you're in your group, you're going to pick one of these to focus on, look this stuff up, and report back to your group. Now, if your group gets finished and everybody's gone around and y'all have said what you need to say about each virus, then you can return to the main room. So I know that groups are finishing up. I may interrupt you either way, just in the interest of time. I'm now going to create your breakouts, and you're going to go away and talk to each other about what you have found for each one of these. I hope that was productive. So what I'm going to do now is, assuming that you had a chance to talk through these different viruses with each other, given that you were in groups of 5, each we just didn't have enough for 6, there may be one of these viruses that you didn't get to. So your answers might not all be the same, but I'm going to ask you a series of questions. And based on the data that you accumulated together, you'll do your best to answer the questions. I expect everyone to answer. If you are asked a question that you can't answer because you did not get to a particular virus, you can always put a question mark, but you should put something into the chat as long as you are not driving. All right, so of the viruses that your group was able to look at, what virus was the largest? Put that into the chat. You should have, and if you don't remember, that'd be fine to put a question mark, but just put something so I know that everyone's had a chance to answer. What virus was the largest based on what your group looked at? Yes, so Ebola is the largest. Now, if you didn't get to Ebola or you didn't remember that, it's not a problem. Again, you're not trying to memorize these. What we're trying to do is take these features that we've been understanding viruses have and apply them to things we know about to make it sink in that this is something to pay attention to. Which virus had a DNA genome? There was only one. If you did not encounter it, you could always say none, because maybe that wasn't the one that your group focused on. But which of the viruses had a DNA genome? Great. And if you don't remember, putting a question mark is fine. Yeah. So it was smallpox. Smallpox had a DNA genome. Everybody else was RNA. I asked this question to illustrate this. I think the tendency for people is to sort of assume that there's like 50-50 DNA, RNA, or maybe like more DNA because that's what we're made with, like our genome is DNA. But there's quite a lot of animal viruses that are RNA based. Which of the viruses was naked with no envelope? Again, there was only one. Maybe you did not get to it. Maybe you don't remember. Whatever your answer is, put it into the chat or put a question mark if you can't remember, but please do answer the question. Which virus was naked? It's very possible you did not get to this one. Okay, so possibly nobody got to this one. It was the rhinovirus. It's the causative agent of one of the forms of the common cold. And I ask this question because, again, I think that the tendency is for people to assume it's like the viruses are 50% of them are naked, 50% are enveloped. There's a lot of enveloped viruses, a lot. Now, your answers are going to be different than everybody else's, but if you were able to find a picture and describe the shape of it, how would you describe your virus's shape? You know, some of them are going to be straightforward. They're just going to be, it was a polyhedron. Or it was a thread or something like that. But others have a distinctive shape you might have discovered. So put whatever you found into the chat. Yes. So those of you who looked at Ebola, you discovered that it had kind of like a U shape, or like looked like a number 6. So it's a helical is the formal name for it. It's like thread-like, but it often has a kind of crooked, like bent over shape to it. Yes, many of you found that your virus was just spherical, polyhedral, whatever you want to call it, that sort of geometric shape that looks like a geodesic dome. Smallpox is typically described as being brick or dumbbell shaped, and rabies is shaped like a bullet. Good, yeah. So here are just some of the ways that these are described. Ebola is like a crooked walking stick, or some of you said it like a number 6. Rabies shaped like a bullet, smallpox like a dumbbell or like a brick, and the others were all geodesic domes. Now, these pages you do not have to memorize by any means, but they're here because I asked you and you might be like, oh, now I'm curious. I want to illustrate that there's all this information now about these viruses that if you were to look at before today, you might be feeling a bit overwhelmed, like I don't know how to make sense of any of this. But now you have a little bit more of a sense of how to understand this information. You can look at the size, it's always going to be in nanometers. The shape may or may not have a distinctive shape, or might be just polyhedral or isometric or geodesic dome or spherical, however they was described. Is it naked or enveloped? Now you know it's got a phospholipid bilayer. What is its genome made of? You know what some of the options are, right? And how many genes? Oh, tiny number of genes for rabies, shockingly small. And what is a spike, right? It's the thing sticking out that's going to bind to the cellular receptor. Smallpox we can see is enveloped, we can see its shape, we can see it's a bigger size, bigger genome, has multiple spikes. What does it bind to? It binds to all these things, right? The common cold, right? Another the naked virus, it's RNA based, it's got this teeny tiny size, it's really small for a virus. Ebola, thread-like, enveloped, helical, it's got a big size. It's a negative sense RNA, and it's got only 7 genes, which is tiny. It has a glycoprotein spike that binds to this particular receptor that's found on most epithelial cells. This explains why it has devastating impacts on your body, because it's infecting like all of the epithelial tissues, basically. Okay, so we're going to round things out by talking about things that aren't viruses, and we will save this for not Thursday, but Tuesday, sorry, the replication of animal viruses. And I have something that I need to tell you about how to prepare for that. So let's talk about these other infectious agents. So in this group of viruses, in this category of microbes, I should say, viruses are one of 3 non-living agents that are non-cellular, but nonetheless can replicate. They're infectious, they cause disease. What are those other 2? So there's viruses, and then there's and Ma. What are the other 2 non-living infectious agents considered in this part of the class? If you don't remember, you can put a question mark. Yes, good. All right, so the other 2 things we're going to discuss today are viroids and prions. Let's start with viroids. What are they made of? What are they made of? Again, if you don't remember, put a question mark. Yes, they are made of RNA, a little tiny piece of RNA, a fascinating tiny piece of RNA. It's so small, it's the size of a small gene, but is not actually a gene, does not code for protein. It does its thing all in the form of RNA, doesn't get made into anything else. All right, true or false, viroids only infect plant cells. Is that true or false? Here you got a 50-50 shot of being right, even if you don't know. So why not take a stab at it? Good job, everyone. This statement is true. So far as we know, the only viroids that are known to cause disease are ones that infect plants. And prions. Now, what are prions made of? Viroids are made of RNA. What are prions made of? Good job. That's right. One protein, and in its infectious form, it's one misfolded cellular protein. So it's a protein's already in your body. If it's misfolded, it causes disease. True or false? Prions only infect animals. And just for the record, when I say animals today, right now, or on a test, I do include people. And if you didn't know that you were an animal, now you do. I do include people in the animal kingdom. Don't be afraid to be wrong. Take a stab at it. You have a 50-50 chance of being right. When are the odds in the real world ever this good? Never. Certainly never when you're buying a lottery ticket. When are the stakes so low? Never. So this is another true statement. In fact, it only infects animals with a nervous system because it infects the neurons. All right, so here is one example of one of those. This is potato spindle tuber disease. It infects potatoes. Here's a healthy potato. Here's an infected potato. It causes stunted growth. The causative agent is an infectious RNA. What are we talking about, guys? Yeah, this is a viroid, and this is a different disease. Spongiform encephalopathy is caused by, this is caused by what? What is, here we see a control, like a normal human brain. Here we see an affected brain where lots of the gray matter has been killed off. This is indeed prion disease. Okay. Now, to prepare for next class, it is vitally important that you do the prep for this. Like, I cannot overstate what you need to do. First of all, if you have not yet watched the video lectures for viruses, make that a top priority. There's a worksheet as well. You want to complete that worksheet so that you have a good understanding of some basic concepts we're going to lean on. And then there is a strip sort activity that you need to bring, and I'm going to show you the course shell right now so we can just locate all these items and then make sure that we know what to do. Let me share my screen. Okay, so here we are in the modules. Here we are in week 7. So make sure that you have watched the video lectures in the prep for Monday section here. There's video lectures, watch those. And then make sure that you are on Wednesday, or not Wednesday, Tuesday. I'm sorry, guys, a little tired right now. Tomorrow, complete and bring the worksheet, and then bring this. Now I'm going to show it to you so you can I can help you understand what it is. At the very least, I want you to print this off and bring it with you, or have it up on your screen, easily accessible. But printing it'll be better for you. This is very much like the strip sort activities we've done for other things, okay? But it's just more options. So we're going to look at 5 different viral life cycles and talk about the steps in those life cycles. And these are all possible steps in those life cycles. Just like with the previous strip sort, not everything happens in every life cycle. So only a handful, like a particular virus life cycle might only require 4 of these steps. And you have to decide which steps in which order for each viral life cycle. I do cover these viral life cycles in the video lectures. What you're going to be doing in class with me is, after we talk about what the life cycle looks like, you're going to try to assemble it from these strips. But let me also show you one other resource that I think is super helpful. So I draw a lot of pictures. You should draw a lot of pictures. But a student with much more digital savvy than I have made a beautiful version of this that you can just steal. Okay, this is the viral life cycle PDF that she made. And what you can see is that she drew out the viral life cycles of the different viruses that we're going to talk about. So single-stranded positive sense RNA viruses, single-stranded negative sense RNA viruses, double-stranded RNA viruses, DNA-based viruses, and retroviruses. Okay, so this might be handy as you prepare and then review. All right, let me go back to our slides. So we have 7 minutes left. I don't think that I'm going to do the genetics Kahoot in 7 minutes. So what I am going to do is let you go a little early, but if you have questions about anything, this material or the upcoming exam, which is next Wednesday, or your unknown ID project, then stick around. If you are done, put your name into the chat to sign out, and have a wonderful night. And if you have questions or need help with anything, then let me know and stick around.